Hello everyone, so in this video we are going to discuss about the MCQs of polymerase chain reactions. So let's begin with the very first question. PCR stand for. The full form of PCR is right option is A, polymerase chain reaction. Okay, so it is a method to amplify DNA. If in case you have a very small quantity of sample of DNA, then you can amplify this DNA sequence. Okay, now come to the second question. The enzyme used in PCR is. So the enzyme used in PCR, of course you are going to magnify the DNA. So polymerase enzyme is required. Yes, so option A is there. DNA polymerase is required for the synthesis of new DNA strand during the PCR. Question number 3. The DNA polymerase commonly used in PCR is derived from. So DNA polymerase commonly used in PCR derived from the take bacteria. Okay, the take polymerase from Thermus aquaticus is heat stable and ideal for PCR. Okay, question number 4. The first step of PCR. So first step of PCR is denaturation. Okay, so denaturation separate the two strands of DNA by heating. Question number 5. Annealing involve. So annealing involve the option A is correct, binding of primer to DNA template. Okay, so primer attached to the single stranded DNA during the annealing. Okay, question number 6. At the typical temperature for denaturation is. So typical temperature for denaturation is 94 to 98 degrees Celsius. Okay, this is denaturation temperature. Okay, then question number 7. The temperature of primer annealing is usually. So the temperature for primer annealing is usually a temperature that is 50 to 65 degrees Celsius. Okay, so temperature for denaturation is 94 to 98, and temperature for primer annealing is somewhat lower, depend on the primer design, that is 50 to 65 degrees Celsius. Question number 8. Extension of new DNA strand happen at. So extension of new DNA strand happen at option C is correct, that is 72 degree Celsius. Okay, take polymerase optimally extend DNA strand at 72 degrees Celsius. Question number 9. Primer R. So primer R option B is correct, short single stranded DNA sequence. So primer are short single stranded sequence complementary to target DNA. Now come to the question number 10, that is PCR can amplify. Okay, so PCR can amplify DNA okay, directly. Question number 11. For amplifying RNA, PCR must be preceded by. So option C is correct, that is reverse transcription. RNA is converted to DNA using reverse transcriptase, that is RT-PCR. Okay, so now come to the question number 12. Which of the following is not required in PCR? So DNA template is required, DNA polymerase is required, RNA primer is not required, and DNTP is required. So DNA primer, not DNA primer, option C is correct, RNA primer, okay, are used in are not used in PCR. Question number 13, that is if real-time PCR, in real-time PCR the amplified DNA is detected. So option B is correct, in real-time PCR the amplified DNA is detected during the reaction. So real-time PCR qPCR monitored DNA amplification as it occurs. Question number 14. What is the role of DNTPs in PCR? So its right option is C, building block of DNA synthesis. DNTPs is act as building block for DNA synthesis. DNTP strains for deoxynucleotide triphosphate are used to build new DNA strand. Question number 15. The machine used for PCR is called. So it is known as thermal cycler. Okay, option B is correct, thermal cycler automatically change temperature for PCR strips. Question number 16. How many copies are produced after 30 cycle of PCR thermotically? Okay, so option D is correct, 1 billion copies can be produced after 30 cycle of PCR. Okay, Now come to the question number 17. Nested PCR involved. So nested PCR involved with 2 set of primer and 2 round of PCR. Okay. Question number 18. Which type of PCR uses fluorescent dye to monitor amplification? So its right option is C, that is qPCR, real-time PCR. Okay. Then come to the question number 19. In PCR, magnesium ions are important because they act as a cofactor for DNA polymerase. Option C is correct. Magnesium ions act as a cofactor for DNA polymerase. Now come to the question number 20. What happens if annealing temperature is too high? So no primer binding will take place if temperature is high during the annealing. Okay. So now question number 21. What happens if the annealing temperature is too low? So if the annealing temperature is too low, option C is correct, that is non-specific primer binding will cause. Question number 22. PCR contamination mainly lead to. So option C is correct, false positive results. The contamination can cause amplification of unwanted DNA, leading to false positives. Now come to the question number 23. A variant of PCR used to quantify RNA level is. So option A is correct. RT-PCR, that is reverse transcriptase PCR, convert RNA to DNA and amplify it. Okay. So option A is correct, RT-PCR. Question number 24. Multiplex PCR allow. So option B is right. Multiplex PCR allow amplification of multiple targets simultaneously. Question number 25. What is START PCR is used to? So its right option is B, to reduce non-specific amplification by activating the polymerase only at high temperature.